these slides are set up for both today and for Monday. And both of those days, we're going to be talking about the phenomenon that immunologists refer to as hypersensitivity. Hypersensitivity is really the fancy immunologist term for allergy. And I can't look at this slide because it will make me sneeze. Because my allergies are just that bad. So in general, there are a type of responses that people think of as allergy responses. In reality, with a hypersensitivity response, there are 4 types of hypersensitivity responses. The ones that you generally think of are probably the type 1s, although we will talk through each of those, these 4 types between today and Monday. Some people can get a little bit sort of generally confused between hypersensitivity reactions and the autoimmune reactions that we were talking about last time. As far as immunologists are concerned, an autoimmune reaction is a situation where the antigen is a self-antigen. You should not make a response to a self-antigen. In the case of a hypersensitivity or an allergy, the antigen is a foreign antigen, but it's a harmless foreign antigen that you don't want to make a response to. So it might be like pollen, for example. So it's sometimes referred to as a foreign innocuous antigen. So I am not a tree, and so my responses to tree pollen are not autoimmune. They're not anti-self. They are against something that's foreign, but it's something foreign that I should not be making a problematic response to. When we look at the 4 types of hypersensitivity responses, they are different based on what is the main type of immune response that's causing the problem. So for 3 of them, there's some type of antibody that's really causing the problem. You can see that the antibody types can change, but in those 3, there's some antibody, and we'll again we'll talk through those specific details, where in one of the other types, T cells tend to be the issue. And then the specific mechanism also will vary between each of them. And then I will also give you some examples for each of them. One reason why it's really important to think about these 4 types of hypersensitivity responses is that if you understand these 4 general sort of problematic immune responses, we can take some of the autoimmune diseases that we talked about last time and match them up. It's actually the same problem. It's a slightly different kind of antigen, but it's the same actual immune response causing a problem. And when we talk about transplant rejection responses, which I think is next Friday, maybe next Wednesday, whatever, it's coming up. When we talk about transplant, there are going to be 3 types of transplant rejections, and they also match up with some of these mechanisms. And so if we get these 4 mechanisms really well, then it's going to make sort of thinking about a lot of problematic immune responses easier. So we're going to start with the first of our hypersensitivity responses. Happily, the first type is called type 1. So we're talking about the type 1 hypersensitivity response. Sometimes this response is known by another name, and that other name is an immediate type hypersensitivity response. There are 2 critical things to say about the naming as the immediate type hypersensitivity response. As you'll see on the next slide, the idea of this response being immediate should get a little asterisk by it. We'll deal with that on the next slide. But the immediateness, and again, we're going to get into exactly what that means, is one of the things that sets this one apart. And so if you are looking at some type of response, and you're going to see, say, you get a bee sting, and you're responding sort of minutes later, that is going to tell you it's an immediate type response, and it's in this type 1 class. And so one of the ways that you can usually tell a type of hypersensitivity is a type 1 is because it's really fast. It's an immediate type. So the key mediator in a type 1 hypersensitivity response is an antibody of the IgE isotype. When you make an IgE dominated antibody response, you have a lot of IgE antibodies. Surprise, surprise. And they get secreted, and those IgE antibodies don't tend to float around loose in the serum. Instead, they all bind to Fc receptors on mast cells. And so your mast cells, like right now, are all coated with whatever IgEs you've ever made in your life. And so it's sort of like your mast cells almost have a bunch of antigen receptors on them. They're not actually the mast cell's own proteins, it's that the mast cell has sort of picked up whatever IgE antibodies were in your body. If you go way back to the material from exam 1, where we talked about the different isotypes, one thing you will notice is that we always said that the IgE levels in serum were really low. That's because it's all sticking to mast cells instead of hanging out in serum. But what that means is that if the antigen were to come around, we would have these antibodies on the surface of the mast cells sort of ready to go. And that antigen could bind those antibodies, and that could induce proximity of those Fc receptors. That could actually cross-link and bring them together and induce signaling. You don't have to do more VDJ or more development. Mast cells don't really need much for activation. In fact, the mast cells already have a whole bunch of granules inside of them, and as soon as this happens, the mast cells immediately release all of those granules. And so when we have antigen that cross-links those IgEs, that will release all of the different mediators that we see. And the specific symptoms that we see can vary with all of the symptoms here being potentials, and I'll talk a little bit about what makes those different as we go forward. But if you look at this, you might realize, like I said, that we have to put a little star by the idea of this being immediate. So if we look at it, we can say, well, it's immediate because the mast cells don't need any prep. They don't need more signals. They already have the granules. The antigen comes, it cross-links the mast cells, boom. The mast cells are releasing stuff. And so that's pretty immediate. However, there is actually an earlier step that had to happen in order to allow this to work. Before you can have that mast cell response, you first need to have a primary response to your antigen that induces class switch to make IgE antibodies. And so you generally do not see one of these types of responses the first time you encounter this antigen. The first time you encounter the antigen, like this lovely pollen, that may go to an antigen presenting cell, that will turn on a T cell, you'll make IgE, and that will bind to the mast cell. And so actually that first time that you encounter the pollen, nothing happens, you feel fine. You're just coating up your mast cells for later. However, the second time you encounter that same pollen, you now have pre-coated mast cells that are ready to go. And now within minutes, maybe within a couple hours, but this is really pretty fast, those mast cells are going to degranulate, they're going to lose their granules, and we're going to be making all of our mast cell products. And so this process is immediate the second time you see antigen, and the third, and so on. You could also imagine how it might get worse the more times you see antigens, because each time you're going to be sort of making more T cells and more B cells and more IgE. And so each time you see the pollen, you might make this whole thing a little bit worse. And then you feel bad like me. And so, in fact, as I've moved around the country, usually the first year I live somewhere, I'm okay, because I'm sort of sensitizing to that pollen. This is sometimes referred to as a sensitization stage. And then in later years, I feel terrible because I've now sensitized to that pollen and have some happy or unhappy IgE. In this slide, as well as in the previous slide, we see the mast cell degranulating and making mast cell products. And so we also need to talk about what are those mast cell products and what exactly are they doing. You can see some of the mast cell products here. Some of them I'm mentioning because I want to talk about them. There are some that I'm going to point out on this slide because you will now start to notice them when you watch commercials for medicines. And you will start looking and be like, oh yeah, that. And so I'm going to point out some of these, even though I probably won't really ever mention them again, so that when you actually start looking at like some of your allergy meds, you'll be like, oh, that's what my allergy med is doing. One set of things that are in those mast cell granules are some toxic mediators. Specifically, the most common one is histamine. You can see histamine right here. Histamine will have a whole bunch of physiologic effects, which I will talk a little bit more about on the next slide. There are a whole bunch of cytokines that are produced by those granules. There are also some enzymes that are in those granules. Those enzymes will actually, you can sort of think of them as being great because they could break down maybe parts of a parasite, but they also can break down parts of the extracellular matrix and sort of wound your body. And we'll see a little bit of that as we go forward. And there are also a number of them that are lipids. So we will see lipids like prostaglandin, which you can see here, and leukotrienes. Sometimes you'll hear them as leukotrienes. I've heard people pronounce it both ways. Both of those things are made from this original lipid, arachidonic acid. And so arachidonic acid can eventually be turned into prostaglandins or leukotrienes, which are going to have a number of different effects as well. One of the reasons why I mention this is that one of the things that you can see here is that to go from arachidonic acid to prostaglandin, there is an enzyme called cyclooxygenase, or COX. COX is the main target of salicylic acid, aka aspirin. So that's what aspirin does, is block that. You can sort of see a little bit of how sort of some of those meds you might take are actually reducing an inflammatory process. One reason why I show you this is that how many of you have ever heard of the allergy medicine Flonase? Yes. So what's Flonase's like stuff that it says in their ads? What's Flonase say that makes it special? Yeah? It doesn't, no, it's not that it has enzymes. So what Flonase tells you, Flonase combats 6 important things in allergies, and 6 is greater than 1. And you will look, if you even look on the package of Flonase, it has the like math of 6 is greater than 1, because it blocks 6 things instead of 1. Yeah. 1, 2, 3, 4, 5, and I think they count this as 2, but that's a 6. That's their 6. So and in fact, if you look really carefully or you go on their website, they have like 6 little like hexagons that tell you the things, and it's those things. This actually shows you what those things will do. So things like our histamines are largely going to do things like induce vasodilation and vascular leak. And so you can imagine those are just things that are going to really help us with an inflammatory response. That's the same kind of stuff we saw with a basic inflammatory response before. And in fact, the cytokines and some of the lipids can also lead to inflammation. The enzymes may lead to tissue damage. And those lipids can also lead to changes in the lungs, so you get some bronchoconstriction. You also may get intestinal hypermobility, so things go out of your intestines fast. You can imagine either direction. You can see that oftentimes, though all of these things together can lead not only to bronchoconstriction, but changes in other muscles, so contraction of smooth muscles in the throat and airways that may lead to problems in breathing. That can also potentially lead to difficulty in swallowing or wheezing. If you contract muscles now in a different location, not in the respiratory tract, but in the GI tract, it's just muscle contraction again, but you get stomach cramps, vomiting, and diarrhea. And so all of those things are just the actions of these mast cell products. Typically, all of these functions that we're seeing can be divided up in terms of how long they take to happen. So some of these responses are known as the sort of more early phase responses, while others are known as the more late phase responses. And so the early phase responses are largely sort of the responses that are happening sort of relatively quickly in terms of sort of your muscle contraction and your expelling contents and sort of starting, all of these sort of more anatomic types of changes, so sort of those changes in function and things like that. But if you actually wait till later, the cytokines particularly take over, and you start to see a much more kind of inflammation-biased response as you're starting to see this late phase response. And so here you can see somebody who's having an allergic reaction. You can see immediately afterwards there's a little bit of swelling, and you can see that, and sort of that can be easily imagined from things like vascular leak and changes in vasodilation, so that we're getting more things going to that area. We get a little swelling. Whereas later on, that person is going to have sort of a more generalized sort of cytokine-based inflammation type of response. We've sort of thought a little bit about some details regarding allergy previously. And so remember that both the environment, and that can include the stuff we talked about regarding the hygiene hypothesis, can play a big role in terms of whether one is predisposed to allergy. So we can think about, all sorts of environmental factors that could either sort of push one towards allergy or protect one from allergy. There are also a fair number of genetic factors. Specifically, when people talk about allergy genetics, they frequently will talk about a condition called atopy. Atopy is something where individuals have a predisposition to making excess Th2 responses. Th2 responses are going to induce things like class switch to IgE, IL-4, that nice mast cell response. And so people who have atopy or who are atopic tend to predominantly make Th2 responses and tend to have a lot of Type I hypersensitivity responses. And so you can see patients who will have asthma and dermatitis and allergies and sort of multiple different Type I hypersensitivity diseases together. And that does seem to have something to do with specific genetic susceptibility being a big part of it, as well as potentially some issues with environmental exposures. And in fact, sort of the Northern European super pale skinned people often are known as atopic. And basically, the easier way to describe that is to just say, I am the poster child for atopy. And if you could think of a type of atopy, I got it. Yes?
So back on the other side, you were talking about hygiene hypothesis. The viruses and also the food particles that cause allergies. What would they kind of go against the hygiene hypothesis then? Because if you do a clean environment, you wouldn't have these viruses and bacteria, so you wouldn't get this
Right, so those are predisposing. So the hygiene hypothesis says that sort of dirty environment. Oh, wait.
Yeah, so that's my thought. If you have a dirty environment, you have more like virus and bacteria. So would you have more?
Where? Wait, I'm confused. So allergens, viruses, and bacteria are sort of influenced by either of these things, and so these things could alter which allergens, viruses, or bacteria are present in your body. Is a big part of it. Okay, does that make sense? Okay. And if we actually look at what some of the genes are that tends to be important in people with type I hypersensitivity, one of them is MHC class II. Surprise, surprise. We also can see variants in the T cell receptor, in IL-4, in the IgE receptor, in some of those leukotriene production things, so genes involved in tolerance and activation and transcription factor. Lots of things you've heard of might potentially be mutated or altered in these types of processes. There are others. So we might see some that are involved in other processes like cell death. You can also see, for example, in asthma, ADAM3 is a pretty important gene which is involved in airway structure. And so you can imagine, and in fact, if we were looking at skin allergies, we would see some skin gene polymorphisms as well. Yep.
What's NFAT C2?
What's NFAT C2? So it's one of the variants of NFAT. Yes, there's variants of a lot of things we've talked about, but so we can also talk about what a type I hypersensitivity looks like, sort of what type of antigens, what the actual symptoms end up being when you have a type I hypersensitivity. So this list lists some of the things that people tend to think about as big type I hypersensitivity antigens. So you can see that all the pollens are good type I hypersensitivity antigens. You can see that people who might have an animal allergy or latex allergy, oftentimes this is a type I hypersensitivity. Some drug allergies. Some of the really severe food allergies like nuts or seafood, eggs, milk, all of those can be type I hypersensitivity. And there are also a number that are insect products. So a type I hypersensitivity is the bee sting reaction. So if you're allergic to bee sting, that's a type I. If you are allergic to dust, that is generally a type I. Just so you know, this always like freaks people out. If you're allergic to dust, you're not actually allergic to the dust itself. You're allergic to these little tiny microscopic invertebrates that live on the dust that are called dust mites. That's one of the dust mite. You're actually allergic to some antigens that are a part of the dust mite that live on the dust instead of the dust itself. This is also the other really big one is this is also a nickel allergy. Can sometimes be a nickel allergy, which I'll talk about nickel allergies later. Yeah.
How small are we talking? Like, how small are dust mites?
Very small.
No, like as in like, you know, nanometers or like an actual like unit.
I don't know a unit, but I'll give you something that'll just make everyone super happy. Your face is covered with them.
Stop it!
My face is also covered with them, yes. They can eat skin cells. In fact, dust is a lot of dead skin cells. The actual details that you see are, and sort of what you see in terms of the symptoms can vary in response to these type I hypersensitivity antigens. And that variation is largely based on which anatomic location comes in contact with the antigen. So if you have something that comes in under the skin, you have one type of allergy called a Wheal and flare. If you inhale it, then you have sort of nasal issues. If you eat it, you have sort of food allergies. If you get it into the blood, then you have anaphylaxis. And so all of these are actually pretty much the same response. It's just that that same response is happening in a different anatomic location. So the first one I want to talk about is the Wheal and flare, which is a type I hypersensitivity response, but one that's happening in the skin. So one that's happening sort of right under the skin. Subcutaneous is right in the layer under the skin. So with a Wheal and flare, you have mast cells that live locally under your skin. If the antigen is present, that will lead those mast cells to make their mast cell products, and that will alter the blood vessels just in that local area of the skin, and you'll get sort of swelling and a little bit of an itch. This is, what you see here is an example of what's known as a patch test. And so if you were to go to the doctor and say, I need to figure out what I'm allergic to, they will put little bits of different antigens under your skin across your arm. So this is somebody's arm. They'll have a negative control. And you can see with the negative control, there's no swelling, no response. They'll put some histamine in as a positive control. And you can see this is sort of what an allergy response looks like. And then they'll put in all sorts of different things. And so you can see that this person is a little bit allergic to plane tree pollen, and I don't know what plane trees are. That always confuses me. More allergic to birch pollen, more allergic to grass pollen, somewhat allergic to daisy pollen, not terribly allergic to mold, not terribly allergic to sheep wool, very allergic to horses, very allergic to dogs, very allergic to cats, and very allergic to feathers. And so you can see sort of that kind of a process. Sometimes there'll be situations where people will have to have both arms because they're having such big responses. There are also situations, there's a really disgusting thing online about this, where there was a person who had such bad responses. They did their arms and the whole thing was like so swollen that they couldn't distinguish them. And so they ended up having to do their whole back and like their whole, yeah, and it's like all over, and it's bad. And I used to joke about being allergic to everything, and then I saw what their back looked like, and I'm like, oh, I guess I'm actually not allergic to everything because that's what it looks like. So that's sort of the idea of a patch test, is basically it's just a little thing where we're seeing, do you make a skin reaction to these types of things? Now, obviously, pollen you might normally encounter through inhaling. In this case, you encountered it by having it put under your skin, and that's why you see the Wheal and flare response. Yep?
Would they ever do like allergies? Because I know my brother's allergic to peanuts, and when he got it on
Yeah.
his back.
Yeah. Yeah.
Okay. And it reacts the same way.
Yeah.
Even though it's not like.
Yeah, because what happens is the IgE is going to go everywhere and bind to all the mast cells. So, and you know, this one is not so serious as potentially that if he ingested the peanut. Yeah. Yes, Mark?
Why is it like less serious if you get it like subcutaneous as opposed to like?
Because you're less likely to get that antigen to go systemically, and because if you have sort of this same kind of muscle constriction happening in your GI tract, it can involve, if it's more actually the problem of if you happen to inhale some of that peanut allergen when you're eating it, it can lead to changes in the lung structure, like, you know, breathing issues, and that's really serious. So it's either if it gets in the bloodstream or if it gets to the lung is where things get particularly serious. Speaking of which, if you have a type I hypersensitivity reaction in the respiratory tract, so if you breathe something in, whether it's pollen or whether it's something from a dust mite, typically the dust mites have these weird proteases, and people have responses to the dust mite proteases. The most famous one is called DERP1. So you'll inhale that antigen, it'll hit the local mast cells, that will induce changes in the blood vessels in the mast cells, blood vessels in the respiratory tract, as these are respiratory mast cells. So we get sort of swelling, and we get vasodilation, etc. in the respiratory tract. We also will get mucus being expelled when these cytokines and these lipid mediators are produced. That makes your lungs and produce more mucus. It usually also induces changes in the muscles so that your airways basically try to expel contents. So you get coughing, sneezing, expulsion of phlegm because you're trying to get it out. by expelling things. You may also see contraction of smooth muscles and constriction of that throat and airways, which can give you difficulties in swallowing or breathing. And so this is why this one can be particularly dangerous. This is also what is seen in patients with asthma, which is a type of respiratory type I hypersensitivity response as well. In asthma, so this is a normal bronchus. There's white space here. That white space is where the air goes in the breathing. You can also see that there is sort of a layer of connective tissue, a barrier around it, as well as additional cells. As mast cells start producing their products, we will start to see vasodilation and sort of that general early response that we talked about. But as things keep going, as we see more cytokines, we're going to see a lot more mucus production, and we're going to see a lot more cell infiltration as part of that inflammatory response as chemokines are calling in the cells. And in the end, what you can see is that there's big changes to the wall around the bronchus because of all of those enzymes that are being made that might be involved in sort of tissue remodeling. We're going to get major structural changes. You're going to see that there are, and so like look how thick this is. There are all sorts of white blood cells that come in, and in fact so this is actually a smaller area. It's full of cells and it's full of mucus. And so now what you can see is there's not so much white space, and so there's no place for the air to go with breathing. And so our patient may have a lot of breathing difficulty at this point. If we see the same types of responses in the GI tract, then that ends up being food allergy. So again, it's still just the antigen triggers the local mast cells. It's just that now those mast cells live in the GI tract. And so the muscles they're acting on are the GI tract muscles. The blood vessels they're acting on are GI tract blood vessels. And so as a result, we get muscle contraction. That usually involves changes in both fluid secretion and peristalsis, aka expulsion of GI contents by diarrhea or vomiting, and stomach cramps, all coming from that muscle contraction as a result of these mast cell products. What you will see here is that it also mentions that sometimes the antigen can start diffusing into the blood vessels. When it does that, you start to see skin symptoms as well. You start to see the antigen triggering skin mast cells. And what happens is that you get urticaria. Urticaria is a fancy word for hives. And so if you have that antigen starting to diffuse into the blood vessels in the GI tract, then you can start to end up with hives. And so these are all of the responses that you're going to see with the food allergy. So this sort of goes through a lot of the same details of this in a table from your textbook. And so you can see that with food allergy, you can see some foods that end up really having sort of the hives, because the biggest place where you start to see the symptoms is when they diffuse into the blood. Others become a bit of really start to trigger mast cells when they're first eaten, and so you end up with sort of itchiness and sort of mouth swelling. You can see the GI effect, but you could also imagine if you happen to inhale something while you're eating, you could see some other types of reactions. And if we get reactions in the blood, then we can get to something a little bit more serious. So we can end up seeing a lot of different responses to those food allergens. You can see that process happening here as well, that we might, if we first encounter an allergen through the skin, we might turn on a Th2 response in this case, or we might turn on some kind of inflammatory response. And then if we later see that antigen in the intestine, we might have sort of an inflammatory type of response. And so the idea that I want to get at here with this slide is that perhaps if the first time you ever saw that antigen was in the intestine, perhaps you would have oral tolerance happen. But if you first saw it, say, in the skin, you would induce an inflammatory T cell response, and then you might end up with the food allergy whenever you eat that as well. And this is the thing I mentioned about how there is this idea perhaps that some peanut allergies are related to lotions with peanut antigen, such that the first exposure is actually a skin exposure instead of a GI tract exposure. Yep.
So, do you know anything about like how prolonged this exposure would have to be, or like repetitive or anything like that, or could it even just be like one time you get exposed?
I'm not sure. And again, that would be a really hard one to do because you'd have to be able to look at your allergic patient and somehow like go back in time and figure out what antigens they had when. And so that would be a particular challenge. Yep, Emily.
So, in terms of like lactose intolerance,
Mhm.
would that be considered an allergy, or is it completely different?
So lactose intolerance is large. So the first part of it is that you don't have an enzyme to break down lactose. And so it's actually more of a, you don't have the enzyme and then you react poorly to the undigested lactose. I think that the reaction to the undigested is might be an allergic response, but I'm not 100%.
Well, because my brother's allergic to milk.
Okay.
Like, everybody's always like, oh, is he lactose intolerant?
Yeah, no, so those are two totally different things. Lactose intolerance is inability to break down. It's you are missing a very specific enzyme that you can't break down the lactose. And I don't remember exactly what happens with the unbroken down, but that's what the distinction is there. And so the final type of allergic response I want to mention is anaphylactic shock. Anaphylactic shock is the response that we see when we have IgE-coated mast cells responding sort of throughout the body. And so we're seeing antigen in the bloodstream, and we're seeing this response happening kind of body-wide or systemically. And so as a result, we're going to see issues or effects on the heart and vascular system. We're going to see issues in the respiratory tract. We're going to see issues in the GI tract. And so we may see things like swelling of all sorts of different tissues, including the tongue, which can make it really hard to breathe. Issues with blood pressure, because all your blood is going into swelling and thus cannot keep blood pressure up. Problems with oxygen to tissues, problems with heartbeat because your heart is responding to the lack of blood, potentially leading to things like loss of consciousness. You're going to have constriction of your throat and airways, difficulty swallowing or breathing, which don't go together and make sweeving. Smooth muscle contractions, and a whole bunch of GI issues. And so this is sort of that really immediate, rapid response that you think of, that people often think of with an allergy. And this is specifically happening when you see the allergen in the blood. So there are a couple, so you might look at this point and say, okay, this is like a terrible response. I hate it. And you might kind of think a little bit about why exactly we have this response. The general idea is that many of these allergens are cross-reactive with helminth antigens. And so we can see, for example, that there is an antigen from certain helminth worms that also is found in cat dander. We can see that other antigens from helminth worms are in types of pollen. Some of the other proteases are similar to proteases that we might find in other types of things like our dust mite proteases. And so the idea is that this evolved in order to turn on eosinophils and mast cells to secrete things at this great big helminth, which we cannot phagocytose. And so it's toxic. We're putting out enzymes to degrade it. We're doing all sorts of things to eventually kill that helminth. There are some people who argue with this theory and who think that this theory doesn't make sense. And so I just want to point out kind of one alternative that some people have argued here. So if you think about this process and imagine having some sort of parasite infection, it's not like a parasite infection kills you right away. if you get a parasite infection, it's usually a pretty long-term chronic infection. It's not like you get a parasite infection and you die particularly quickly. Yet this hypersensitivity response is the immediate type hypersensitivity response. This is a response that happens within minutes to hours. So why the heck would we evolve a response to parasites that happens in minutes if parasites aren't particularly dangerous in minutes? And if you think about it, many of the effects you see here involve expelling things. You're expelling your GI contents. You are expelling your respiratory tract contents. You are getting stuff out. And so there are others who argue that perhaps these responses first evolved in response to things like venoms or other toxins or other poisons. And so things like bee venom or snake venom also actually cross-react with some of these same types of antigens. And so maybe the idea was sort of, this was your original immune response to sort of poison and venom, where it could hurt you very quickly and you need to expel right now to get out the bad stuff. And this response has sort of evolved in a problematic way. And so there are some type of people who think about allergy in relation to some of these other types of substances instead. And so you can see some of those arguments. So this is sort of the type I hypersensitivity response. IgE and mast cells and histamine are the key parts. And the other key part is it's immediate. If you see someone who's having an immediate type hypersensitivity response, it's happening within a few minutes. So let's imagine that your friend gets stung by a bee and has a bee sting response. Type I hypersensitivity bee sting response. Is that the first time they've ever been stung by a bee? No, why not? Yeah.
Because if it's a bigger response, that means they have to have a lot of it, which means they must have encountered it before.
Okay. What if we don't even think about bigger? You need the IgE class switch. And so the first time with these types of responses, you get sensitized, and then it's the immediate type response in the second and subsequent times. And so if you see somebody making a type I hypersensitivity response, it actually means it's at least their second time seeing that antigen. So to contrast, we have a type II hypersensitivity response. So with a type II hypersensitivity response, again, antibodies are going to be the problem. The antibodies in this case are not IgE anymore. Now, the antibodies are either IgG or IgM. When you see a type III later on, you will notice that IgG and IgM are the problem in type III as well. And so it clearly isn't just about having IgG or IgM here. There's something else that distinguishes type II versus type III. And the other big thing that is important in a type II hypersensitivity response is that the antigen is on a cell. So the antigen is a cell surface antigen. So here you can see it's a green triangle. It's a surface antigen. And that surface antigen is on some kind of self cell. It's bound by some antibody, and as a result, we see somehow this cell get killed. So we're going to have a surface antigen, and the cell is going to get killed as a result. The details of this are kind of best explained by giving you two examples. They're two really famous examples of type II hypersensitivity response diseases. The first one requires me to say something quickly about the antigen. And so the antigen that we are going to see in our first example of a type II hypersensitivity response is a blood group antigen. One thing I will also make sure to notice is with a type II hypersensitivity response, I have not told you anything about timing. So timing's sort of not a big deal here, unlike with the immediate type. where timing is a big deal. Or when we later get to type 4 and it's called delayed type, that one timing matters. Type 2 and type 3 timing isn't a big deal. So blood group antigens are going to be the antigens we see in this example. So we have red blood cells. They have some sugars on them, and there are differences in sort of what the terminal sugar is. So it can be galactose, it can be N-acetylgalactosamine, or it can be nothing. And that distinguishes between A, B, and O blood types. And in the end, you have red blood cells that either have A antigen, B antigen, A and B antigen, or no carbohydrate. You can also have RH or not. In fact, the RH antigen is going to be the one that's particularly important in our example here. But the idea that I want you to see is that this is an antigen that's on the surface of cells, specifically on the surface of red blood cells. The disease that, the example I want to tell you about is called Erythroblastosis fetalis, also known as hemolytic disease of the newborn. Sometimes you'll hear it talked about as something called blue baby disease. So this happens in a situation where we have a woman who is pregnant who is RH minus. If this woman who is pregnant is RH minus and the fetus is RH plus. So here, mom is RH minus, but baby is RH plus, and so baby has red blood cells that contain the RH antigen. There's maybe a teensy bit of those antigens being in contact with maternal circulation, not necessarily a ton, but at delivery, we do see some mixing of baby's blood and mom's blood. And so we do see mom suddenly being exposed to these red blood cells from the baby and having those red blood cells now have this new foreign antigen. And so mom has B cells that respond to that antigen, that respond to RH, and mom starts making antibodies to RH. Everything's fine. However, if mom gets pregnant again and has another baby who is RH positive, now mom has made antibodies, IgG antibodies against this RH antigen. And if you remember, mom's antibodies can cross the placenta. And so now these antibodies will cross the placenta and will actually get to the baby. And those antibodies will destroy the baby's red blood cells, which have the RH antigen on it. And so in the end, the baby is going to be born with no red blood cells. The baby's going to be born with anemia. The dead red blood cells also actually lead to some other problems, which is it's excess bilirubin that end up being part of the reason why the baby is in the incubator. And so you can see that this could be a really big problem. Yeah?
So, do they do any type of testing after the mom's pregnant?
So they do. And fortunately, we now have a treatment for this. And so they do testing, and they give mom the treatment that I'm going to tell you about on the next slide if, in fact, they realize that this problem is going to happen. And so the treatment they give mom is a drug known as Rhogam. Rhogam is pretty straightforward in terms of how it works. Rhogam is actually an antibody. And Rhogam is an antibody against this antigen that is actually an IgG antibody. If they give mom Rhogam during her first pregnancy, so they actually do the testing and do this during the first pregnancy, these antibodies, and you'll see this on the next slide as well, turn off B cells. So mom can't make a primary response. Because remember that if we have pre-existing antibodies, that turns off a primary response. And so mom can never actually develop her own antibodies here and never give them to the baby. And so here you can see that same thing. In the first pregnancy, mom has the baby's red blood cells. They have the Rh antigen. This binds to a naive B cell, signals that naive B cell. In the second pregnancy, you can see that we've got memory B cells present now. We're going to turn on those memory B cells. We're also going to be making lots of antibodies that can cross the placenta. We're going to be very sad. The baby's going to be anemic. If we treat mom during all of her pregnancies with this antibody that binds to the antigen, you can see that it will bind to antigen as well as to the naive B cell. Naive B cell is yellow while the memory B cell is orange. It will lead to a negative signal through the Fc receptor, and so we will never turn on mom's B cell response, and she will never end up with memory B cells that can make antibodies to harm the baby. And so she will have healthy babies. And so this is sort of using what we've talked about in terms of B cell activation in order to do good. Yay. So that's one really famous type II hypersensitivity reaction. We've got the cell surface antigen. Antibodies are the problem, and the cells eventually end up getting destroyed. It can be through complement. It can be through NK cells. We sort of don't care. It's just the cells get destroyed somehow. The other example that I want to give you is an example of a penicillin allergy. In reality, if a person has a penicillin allergy, it could actually be any of the 4 types. So there are type 1 penicillin allergies, type 2, type 3, and type 4. I am going to give you an example of the type 2 penicillin allergies. However, if you hear this and say, that doesn't sound like my penicillin allergy, it may be because your penicillin allergy is one of the other types. So what you can see here in the middle is the structure of penicillin. Penicillin has a bond that is shown here that is reactive. And the goal of penicillin is for that reactive bond to bind to an enzyme in bacteria to stop bacterial cell wall synthesis. So ideally, this bond reacts. It binds to this bacterial enzyme. The enzyme is now unable to work. The bacteria cannot live their lives. However, in some situations, that penicillin reactive bond will react instead with a red blood cell. As a result, your red blood cell will now have penicillin attached to it. This will be a foreign antigen bound to your cell. So you have this modification on a protein on the red blood cell, and that modification is basically a foreign antigen. And so if this is actually what we see happen, then we will have the red blood cells that are modified to have penicillin on their surface. So now we have a cell surface antigen that can be bound by our antibodies. You can see the antibodies binding here. As a result, complement can attack those red blood cells and lyse them, or those red blood cells can be phagocytosed by macrophages and be destroyed that way. In either case, we have no red blood cells anymore. And so in either case, our patient is going to end up anemic because of this lack of red blood cells. And this disease is known as hemolytic anemia. Hemolytic because we are lysing red blood cells, and that's why the patient has anemia. And so you can see this as being a big problem for our patients. So I want to just check one quick thing. All right. On Monday, I'm going to tell you about type 3 and type 4 responses, as well as talking about treatments.